This video is going to run you through Chapter 10, The Molecular Biology of a Gene. It's essentially a chapter about DNA and how DNA works, what it gets used for, what it's made of. It's just everything you never wanted to know about DNA, essentially. The study of DNA is a branch of molecular biology. Molecular biology just studies all the molecules relative for life. These earrings are for super nerdy people like me. These are some of the molecules that are necessary for you to live your day. Like without acetylcholine, your muscles wouldn't contract. And without serotonin, your mood would not be cool. And without dopamine, you would feel pain and you wouldn't be able to move like you were supposed to. This helps you sleep at night. And so all of these things, your genes tell you how to make them. And so both these and the genes that help you make them are part of molecular biology. There was a debate in biology for a long time about what the genetic material actually was. People thought that they were particles or goo or plasm. What we had made it down to in the 40s, 50s, and 60s was it was either going to be a protein or it was going to be a nucleic acid. And most of us thought it was going to be protein because proteins are so large and complex. We just thought that they would be the genetic material, and as it turns out, it's actually the simpler DNA and RNA that gets used for that purposes, and we learned that through experiments like the one that I'm going to describe to you here. The critter that was used to help us figure out that DNA was the genetic material is called a phage. Their full name is bacteriophage. These are viruses that infect bacteria, and they really do look like little alien spaceships. They're only made out of two things: protein, which is everything in yellow in this picture, and then DNA or RNA, one or the other, but this is a nucleic acid, so that's the blue stuff that's in there. So since our debate was between, is DNA the genetic material? I said DNA, so if DNA is genetic material or if protein's a genetic material, this one thing is only made out of those two components, and so it would help us figure out which one actually could transform an organism or control how it was going to be processed. So phages infect bacteria, and when they do that, they transform the bacteria, or they make it behave in a different way. So what Hershey and Chase did was they used radioisotopes to label the protein and to label the DNA, and then they measured which actually got into the cell. Was it the sulfur, radioactive sulfur, which would mean the protein got in and transformed the cell, or was it the radioactive phosphate, which is part of nucleic acids, which would mean that the DNA made it into the cell? And as it turns out, the protein never even enters the cell when a phage infects bacteria, only the nucleic acid does. So what Hershey and Chase showed is that the DNA is what was capable of transforming the bacteria, so the DNA must be the genetic material present inside the cell. I think you guys are filling in blanks on this one, and I think the blanks are the words that are in red, but most of this is typed out for you guys, and so just make sure you get those words in red. So this is just the description of everything that I just told you on that last slide. Remember, you can pause the video so you can get stuff because I'm going to go fast through here. So nucleotide include the chemical makeup. So these are the building blocks that make up nucleic acids. We talked about this the first time when we were doing organic molecules way back in Chapter 2. This whole thing is one nucleotide, and you should draw it in the space that I've provided you on page 1 of Chapter 10. There's 3 parts to a nucleotide. You have a phosphate, which is a phosphorus atom and then some oxygen atoms. You have a 5-carbon sugar. This is the thing that's going to be different between DNA and RNA, because in DNA the sugar is deoxyribose, and RNA the sugar is ribose. And then there's nitrogen-containing bases, and these are the things that make the different A's, G's, T's, C's, or U's, depending on whether you're looking at DNA or RNA. Next up, when you string together a bunch of nucleotides, you get a polynucleotide. RNA works as a single-stranded polynucleotide, but DNA is 2 of these put together through hydrogen bonds. There would be another one next to this. So this is a polynucleotide, where there's 1 nucleotide, 2. When you get 3 or more, that's what poly means, is you get a bunch of those. When you line these up together side by side, what ends up happening is down the side of your polynucleotide, you get alternating sugars, then phosphates, then sugars, then phosphate. And so this creates a sugar-phosphate backbone all the way down the side of the molecule. What are the 4 nitrogenous bases present in DNA? These are the 4. This also shows you which one are pyrimidines, that's thymine and cytosine, and which ones are purine, that's adenine and guanine. This does not show you what's going to base pair with what. We'll come back to that here in just a minute. But this shows you the chemical structure of each one of those. So pyrimidines consist of 1 ring, purines consist of 2 rings, stupid memory trick that somebody told me forever ago. Ags are pure, so they wear 2 rings, and so the purines are 2-ring structure, and it's A and G for ag. What are the 4 nitrogenous bases present in RNA? It's still cytosine, adenine, and guanine, but instead of thymine, RNA gets uracil instead. This is a 1-ring structure, and so this is another one of the pyrimidines. But that's the only thing that's different aside from the sugar, is there's no thymine in RNA. We use uracil instead. Okay, for compare and contrast DNA and RNA polynucleotides, this table summarizes that for you. I've said a lot of this stuff up at the top, so I'll just kind of come down to the bottom. In eukaryotic cells like yours, your DNA is kept inside the nucleus to protect it. Your RNA is made in the nucleus, but then it leaves so that it can go out into the cell and do things out in the cell, so it does leave eventually. Then the function of each one of these is slightly different. So DNA, I like to kind of consider it your cookbook. It's your recipe for every single protein that your cells need to be able to make. Versus RNA, these are intermediaries that help the DNA do its job, but they're not your recipes. Sometimes they're copies of recipes, but sometimes they're helpers. And so RNA has more of a functional role versus this is just the book of recipes. Double helix. When you have the 2 strands of DNA side by side in your double helix, you end up with what some books like to describe as the rope ladder. Down the sides of the rope ladder, you have your sugar, phosphate, and then the rungs of the ladder are your nitrogen-containing bases, your A's, G's, T's, and C's. The problem with DNA is it doesn't exist as a straight ladder like this. Instead, because of the chemistry, it twists around itself. Because it twists, it's considered a helical shape, and it's a double helix because it's 2 strands or 2 polynucleotides of DNA. Rosalind Franklin I usually like to mention because basically I feel so sorry for her. She got screwed in like every possible way that you can imagine. So she was a grad student who was taking X-ray crystallographic images of DNA. Here's what I want you to imagine. Imagine that you were standing at the top of this rope ladder and you took a picture looking down the ladder. What you would see is the circle that was the rope around the sides, and then the rungs would form the X inside of there. That's what she did. She took a picture of DNA down the length of the molecule and she got this image right here. She had left this image out on her table, and a couple of guys that we're going to talk about on the next slide, Watson and Crick, came by. They saw her picture and they asked her boss, the guy who was running the lab, if they could have it. And without ever asking Rosalind if they could have the picture, they took her picture and then created the model of DNA, this model of DNA right here. So they helped to elucidate the shape. They got Nobel Prizes. She died from radiation poisoning from all the X-ray images that she took of DNA and then various proteins as well. So she should get a lot of credit for helping to establish the basic chemical structure of DNA, and yet she was a woman who died doing her science and nobody even bothers to learn her name. So I'll make you learn her name, Rosalind Franklin. There you go. Watson and Crick are the ones who got the Nobel Prizes, and what really extra pisses me off about this is one of them is such a massive, unpleasant person. I'll try to be nice as I make this video. So Crick is dead. Watson is still alive, James Watson. He is such a horrible person that nobody wants to work with him. He is a Nobel laureate that nobody wants in their lab because he is a woefully misogynistic person who never treats women with respect. He still believes that eugenics should be practiced, which basically means he thinks that we should weed out weaker races and try to make them master race. In other words, he and Hitler would have been really great friends back in the day. Because nobody will work with him, he had to sell his Nobel Prize and try to make some money so he didn't starve to death, essentially. So it just goes to show you that sometimes the good people do not end up getting the rewards. The people who exploited them do. Alright, so that's me getting off my soapbox for just a second. Next up, you have explain complementary base pairing. So, but within DNA, you have A base pairing with T, and you can see that here's a base pair, A to T, T to A. C base pairs with G, cytosine to guanine, and you can see that all up here at the top. C to G, C to G, G to C. The problem that most students have is when we go between DNA and RNA. So if this were a sequence of DNA and we were going to be making RNA against it, A would like to base pair with T, but you can't put T's on RNA. You would have to substitute a U there. So A base pairs with U. However, T still base pairs with A because you can still put A's in RNA. C and G still work exactly the same. So the only difference really between DNA and RNA is that there's no T's in RNA. Any place you would have wanted to put a T, you have to put an A there instead. All right. This is sort of a math question to see if you understand that base pairing and how it's going to work. So if adenine makes up 20% of the bases on DNA, pardon me, I meant to hit this button, you should be able to figure out what everything else is going to be because of those complementary base pairing rules. Because A is going to base pair with T. If A is 20%, that means T also has to be 20%. So 20 plus 20, that tells us that the A's and the T's are making up 40% of the bases on DNA. That means the remaining 60% has to go between the G's and the C's. But those are going to be base paired together. So half of that 60 is going to go to G, which means the answer to the question is 30% are going to be guanine, and that's because the other 30% is going to be cytosine. This is called Chargaff's rule, and it just goes back to complementary base pairing. A's and T's are going to be relatively equal because they're always going to be base paired together, and G's and C's are going to be equal because they're going to be base paired together. All right, for the N-class work, it says to grab a whiteboard and to make these sequences. I'm going to attempt to do sort of what would have been on there on the screen. Actually, I'm going to blank my screen out before I do there. Hold on. Like you guys have any choice in the matter, right? Okay. So, it tells you to build a strand using magnetic nucleotides. This would have been our hands-on stuff, but you guys don't unfortunately get that. Oh yeah, I need to get my pen out. So if our strand was A, T, G, C, T, C, A, G, G, T, G, A. If I give you one strand, you should be able to figure out what the opposing strand should be. So what we're doing for this first round is essentially what's going to amount to DNA replication. If there's an A on one strand, it's going to base pair to a T on the other. If there's a T, it's going to base pair with an A. If there's a G, it base pairs with C. And so the opposing strand is going to be the complement to that first strand. See if you can keep doing the rest on your own, and then I'll write them down here in just a second. Okay, so there's how that would have been replicated. So on your blanks under number 4, you would write that in the first blank, second blank, third blank, fourth blank. That's how you replicate DNA. The question under there, why do cells carry that out? We do that so that we can replicate the genome, because if the cell is about to divide, like remember mitosis, during the S phase of interphase, we replicate the DNA. So this process is what happens during that S phase of interphase. So we do that, mitosis, for growth and reproduction. Also just for repair of tissues, should you be injured, you do mitosis for that too. When do they do it? That's during the S phase of interphase. Pardon me. Okay. Explain what is meant by the semi-conservative model of DNA replication. So what this is showing you is this is an original strand of DNA that had already been present in a cell. When DNA replication is going to occur, we have this enzyme that's called DNA helicase, and what it does is it separates the two strands so that instead of being base paired together like it is here, one strand pulls one way, the other strand pulls the other way. Then another enzyme called DNA polymerase comes in, and it adds new nucleotides to the template strands, which are the original strands that used to be there. And then the same thing happens on this one. At the end of DNA replication, you end up with 2 molecules of DNA. Each one of those molecules consists of one old strand and one new strand. So it's called semi-conservative because we conserve the old strand but add a new one to it. So each new molecule of DNA that your cells make It's half old and half new. You have some DNA in your cells that has been there since the moment you were created as a zygote because of the semi-conservative model of replication. All right. When DNA is replicated, oh, I already said that. DNA polymerase is the next thing that you guys have. So there's multiple DNA polymerases. Since you guys are non-majors, we're just going to kind of lump them up together. So this is just the group of enzyme that builds the daughter strand or the new strand of DNA using that old one as a template. Because of a quirk in the chemistry of DNA, as we replicate DNA, we can't actually glue the fragments together like we need to with DNA polymerase. And so we need a whole other enzyme to help us glue those two molecules together, and that enzyme is called DNA ligase. So it just glues some DNA fragments together. I'll show you that one in a video later on in the hopes that it helps you understand it better. This is the book's picture of DNA replication and how that's going to happen. So DNA helicase has done the separation thing right there. The original parental or template strands are in blue. The daughter strands, the new strands that are being made, are in gray. This is a DNA polymerase. It's building the new strand as it reads. Now again, because of this quirk in chemistry, what it means is one of the strands can be built continuously from start to finish. The other one gets built in these little fragments. So we build this one, and then as that fork keeps pulling back, we build the next one. And then we build the next one. And so we keep building it in these little fragments. A second ago I mentioned DNA ligase. DNA ligase is what comes in and it glues these two fragments together. And so you can see that happening there where it's gluing those two fragments together. So you need a bunch of different enzymes or proteins to help you replicate your DNA. All right, here's a video to help show you that overall process and how it's going to work. And I don't know if I can do it without... How do I get off the pin? I'm so sad, y'all. I can't even tell you. I want my pointer back. Give me back my pointer. Alright, fine. I'll do it this way. I'm still going to show you a video. Haha! There we go.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Hopefully that gives you a better picture in your head about how your cells replicate DNA prior to mitosis and meiosis. Next up, genotype and phenotype we defined back in chapter 9 as we were doing genetics, but once again, genotype is the actual genetic makeup and phenotype is the outward appearance of an organism. Essentially, the reason we're coming back to that now is genotype is talking about the DNA that's actually present in the cells, but phenotype is talking about the products of that DNA. Like, what does your DNA tell you to make when you look at a particular gene so that you can look at a trait? Like, when it comes back to the flower color, the reason that a flower can appear purple is because a gene codes for this purple pigment that they produce. And this one doesn't make that purple pigment, so it ends up being white. And so the genotype is the DNA or the gene that codes for the pigment, which is the phenotype. So the steps that are going to get us from genotype to phenotype are involved in protein synthesis, and there's two overall steps. The analogy that I usually give, and I'll give you this on a slide later on, is I want you to again imagine that your DNA is a recipe book for how to make every single thing inside of your cells, and your DNA has been inherited from your great-grandparents to your grandparents to your parents to you. It's important. If you screw this up, you die. So we don't just want that sitting around anywhere in a cell where it can be damaged. It has to stay in the nucleus so that we can protect it, but at the same time we have to use those recipes so that we can make our proteins or our dishes. So anytime we want to make a specific protein, what we do is we copy the one recipe that we want to use right now. That process of copying one recipe from your whole recipe book is called transcription, and what it does is it converts a gene on DNA into something called messenger RNA, or mRNA for short. That mRNA, since it's just a copy of a gene, it's okay if that breaks down. So that's allowed to leave the nucleus, and it's going to float around out in the cytoplasm where it's going to encounter a ribosome. Some of your ribosomes are just floating around in the cytoplasm, and some of them are attached to the rough ER, but what's going to happen is the ribosome is going to be the chef that reads the recipe and then cooks the protein. Proteins are called polypeptides until they get folded into the correct shape, and so this is a polypeptide. It's going to make our folded protein later on. So the steps to making a protein are copy your recipe, that's transcription, cook your recipe, that's translation. So here's what you need to make sure you have written down for transcription. Now, if the, oh, I need to change my thing back to my pen, hold on. If this is a gene that's present on DNA, we're going to do sort of like DNA replication to carry out transcription, only instead of, complementary base pairing with A going to T, A is going to go to U instead, but everything else is the same. So G still goes to C. A wants to go to T, but it can't, so we're going to put a U there. G still goes to C. See if you can transcribe the rest of that, and I'll catch up with you here in just a second. I want to see how y'all do. Alright, so there's our transcript, and I know it's messy. I wish I could write neater on a computer, but I can't. But that's how transcription works. You read the gene, you transcribe it into RNA instead of DNA, so that's transcription. Next up, translation is the cooking step. So you need to have the ribosome. It's going to be the chef that actually does the cooking. But then you're going to have some helpers. So the mRNA is the recipe that we're reading. The tRNA is what I like to call the sous chef. It has the ingredients for making proteins on one end, which if you don't remember from the earlier chapter, proteins are made out of amino acids. So that's an amino acid, cysteine, that's tyrosine, serine, valine, whatever. There's 20 different amino acids. On the other side, it contains what's known as an anticodon, and an anticodon is going to match up to a codon on the mRNA. Now, usually at the beginning of our recipe, I don't know if you guys have ever tried to cook a recipe from some Instagram person or somebody on like Pinterest or something, but they always have this stupid crap story at the beginning that nobody cares about. It doesn't help you cook the dish. But then there's the actual recipe that you're going to use, and then they have the stupid crap story at the end that nobody wanted to hear about either. Same thing happens when it comes to this. There's crap at the beginning that doesn't matter. There's crap at the end that doesn't matter. But in the middle, there's your recipe for actually making something. So we have to know when it's time to start. And one of the tRNAs is going to help us with that. Now, if you flip to the next page in your notes, you guys have a box that looks like this. And I'm looking for the picture. And a circle that looks like this. I'm going to start with the box. Okay. Let's say that your mRNA has the codon AUG, and I'll write that down. It's actually already up there, but whatever. When you use the box, the first base, the A, is going to be on this side. Second base is up here. Third base is on this side. You go to the place where all three of those come together, so AUG codes for MET, which is the amino acid methionine. That's also the start codon. So if I come back to what we had been playing with earlier, gotta find the right slide. Okay. What it means is your ribosome doesn't start reading until it sees AUG. So it ignores. There's the codon that's going to help it start. So what that AUG is going to do is it's going to be translated into the amino acid methionine. After you translate that one, you come down to the next three, CCA. You look that up in either the box using the rules that I just showed you a second ago, or I'll show you how to use the circle this time. Okay, so our codon, our second codon was CUC, if I typed that out correctly. So to get there, you start at the center of the circle and you work your way out. So CUC codes for leucine. And so we would come back to where we were writing in our notes. Ah, darn, I put CCA over here. Okay, hold on. Okay. Nope, that wasn't the one I was on. CCA. CCA codes for proline. Sorry, I have something different typed in my notes than I had on the slide. So, coming back, this one is going to code for proline. So we put PRO because that's the standard abbreviation for proline. Then what happens is the ribosome covalently attaches those two amino acids together with a peptide bond. Then we move down to the next codon, UAC. Now at this point you guys use either the box or the circle, whichever one you feel more comfortable with. What does UAC code for? Okay, codes for tyrosine, TYR. So we'd attach those together. Move to the next one. What does UGA code for? All right, UGA is stop, and stop's not an amino acid, it's the period at the end of the recipe. So what that would mean is we have made a polypeptide out of methionine, proline, and tyrosine, and we put those together. So that's how translation works. Next, explain the one gene, one polypeptide hypothesis. So this was created by Beadle and Tatum. They actually created the one gene, one enzyme hypothesis. We've modified that just a little bit. They worked with mold. This is Neurospora crassa. What they would do is they would hit it with radiation to knock out a gene, and what they noticed is if they knocked out a gene, that mold couldn't make an enzyme anymore. And so they thought that all genes coded for enzymes. Well, now that we know a little more and we've done a little bit more research, it doesn't just make enzymes, it makes virtually everything that your body needs to be able to make, either by coding for enzymes, directly, coding for enzymes that help you catalyze reactions, or coding for other protein products that your cells need to be able to make. And so by saying one gene, one polypeptide, we've backed it out to say they make more than just enzymes. They make a lot of different things. That said, I still do tend to throw back to one gene, one protein. It's just not really realistic. It makes more than that. Okay. Triplet code. All of DNA is written as a series of 3 letters. If you kind of think of it like 3 letters, one word, each word tells you one thing. So on DNA, that's a triplet, that's a triplet, that's a triplet, and that's a triplet. When you transcribe DNA into RNA, we keep things as 3 letters, but the name changes. And the name changes for a very specific reason. That is a codon. It's called a codon because it codes for a specific amino acid. It's still 3 letters, it just has that name change. Codons are also the thing that you have to look up in the box with the circles. So you'd look up that codon, then that set of 3 letters, then that set of 3 letters, then that set of 3 letters. Those code for specific amino acids when you use the box or the circle. The genetic code is the box or circle, whichever one you decide you want to use, and you don't have to memorize those at all. You just have to be able to use them, so make sure you practice using those. We'll do that again here in just a minute. RNA polymerase is the enzyme that carries out transcription, so it's kind of like DNA polymerase. DNA polymerase makes DNA, RNA polymerase makes RNA. mRNA, oh, that was on the last slide. mRNA stands for messenger RNA. This is the copy of a recipe or a copy of a gene that you want to make that can leave the nucleus. But there's 2 other kinds of RNA that are going to help us out. tRNA stands for transfer RNA. This is what carries the ingredients for proteins around. So it has the amino acid at one side, and then it has the anticodon that is going to allow it to match up to a codon. So the codon and anticodon are complementary base pairs using just RNA, no DNA. Next, third and final type of RNA that we're going to talk about is rRNA, which is ribosomal RNA. This is actually part of the ribosome, so it helps with translation. AUG is the start codon. It's at the beginning of every single gene for human beings. It also does code for the amino acid methionine. Be able to use genetic code, and I gave you the box or circle. Again, whichever one you want to use is the one you're allowed to use. Okay, that's later. So next thing that you guys have is another little in-class activity where we're going to do some hands-on stuff. So let me get myself another slide that is blank so that we can practice that. Might as well do it here. Okay, so I gave you a DNA sequence, TAC. Why do I always forget to set my marker back on? TAC, GAG, TCCACT. Okay. First thing that we have to do is transcribe that into RNA when we're carrying out protein synthesis. So when we transcribe, remember we use the rules of complementary base pairing, but we can't put any T's down. T wants to base pair with A, so that one's not a problem. A wants to go to T, but we can't put a T there, so we're going to put a U there, and then that's G. See if you can transcribe the rest of that sequence, and I'll catch back up with you in a second. All right, so there's the transcription. Next, we're going to use either the box or the circle, whichever one you want, to translate that into the amino acid sequence. So these are DNA, and you can tell because there's T's in it, so these are triplets. These are codons. So the first codon we want to look up is AUG. AUG, again, is the start codon for everything, and so that's going to code for the amino acid methionine. Next, look up CUC. What does that code for? Codes for leucine. The ribosome is going to bond those together. Next, look up AGG. That's the next codon. Codes for arginine or ARG. Ribosome is going to bond those together. And then look up the last one, UGA. That codes for stop. Stop means stop, so we're done. So now this is what you would write down, those things under number 5. Okay, the next couple of little questions that I have refer back to mutation. So a mutation is any change in the DNA sequence. What I want you to see is how those could actually affect what's going on inside the cell. Ugh. Me and you, we're going to have problems today. All right. So, what number 6 says is if there were a mutation in the original strand of DNA so that the second triplet read GTG instead of GAG. So here's what that means. Instead of this, it was this. Would the protein be altered? Well, in order for us to figure that out, we have to retranscribe that. So now instead of putting a U here, that would be an A. So then you'd have to look up CAC. What does CAC code for? It codes for histidine, or his. So where our protein used to be met-lu-arg, now it's met-his-arg. The analogy that I usually use to describe that is, let's say that you were going to be making Kool-Aid. Kool-Aid has water, Kool-Aid, and sugar. Well now, instead of putting Kool-Aid in it like you were supposed to up here, you put this in it, and that's soy sauce. So you have put water, soy sauce, and sugar together. Is that still Kool-Aid? Heck no, that's not Kool-Aid. And so that's how mutations change you. It changes the ingredients that you're putting into a protein, and it makes you make an entirely different dish or protein at the end of that because you changed the amino acid. Okay, next step. I'm going to change my color again as we go down to number 7, now that I'm starting to get the hang of this whole color change thing. Okay, if there were a mutation in the original strand of DNA that altered the 4th triplet so that it read ATT, so this time this letter gets changed. Well, remember you have to retranscribe it and retranslate it. So ATT would become UAA. What does UAA code for? Still codes for stop. So does that change the protein? No, stop is still stop. One of the things I want you to kind of notice is, look at, if you had a codon that was UC something, it doesn't matter what that 4th letter is, it's going to code for serine every single time. So generally speaking, if you change the last letter in a codon, it's probably not going to change the amino acid. That creates what's known as a silent mutation. The DNA did mutate, but nothing changed as a result because you still make your dish exactly the same way. You're still adding all the ingredients you were supposed to. That same thing happened here. We didn't change anything except the DNA, but that didn't change you. You have no disease symptoms even though you have mutated DNA. So number 8, do mutations always lead to a change in the protein? No. Silent mutations like that one we just talked about exist. Okay. After that, we have the 6th grade version of protein synthesis. This is that analogy I've been going with this whole time, which is your DNA is your master recipe book for how to make everything that you need to make. You have to transcribe that before you can use it because you don't want to leave your recipes, your DNA, right next to the stove as you're cooking. So you make a copy of it. That's what transcription is. Then the copy is going to go down to be cooked in translation, where the ribosome is going to do its nice little ready thing and then cook the protein. Next step, the little descriptions of mutation. So mutation is any change in the DNA sequence. Like we said earlier, they might cause problems where you make soy sauce Kool-Aid, or they might not, where you get a silent mutation. But a mutation does happen all the time inside your cells. The more you expose yourself to carcinogens or sunlight, which is a carcinogen, the more you change the DNA that's present inside of your cells. Okay. This one's always kind of really tough to talk about, but it relates back to the fact that everything here is written as triplets. So every word that we have in DNA and in codons is a 3-letter word. So I want you to imagine a 3-letter word sentence. So I'm going to say the dog ate the cat. And again, sorry it's so messy. But if we do what's known as a substitution, where like we change one letter in this, that doesn't change the whole sentence. It changes one word, but it doesn't alter the whole entire reading frame. Sometimes though, what a mutation does is it deletes a letter. If we delete that letter, what it ends up doing is it shifts everything down one. So we would have the word the, just like before, but then it becomes DO, and because we deleted the G, an A comes next, and then TETHECAT. That becomes the sentence, and what I want you to notice is that is now gibberish, and it's gibberish because we altered the reading frame. Anytime a letter gets deleted or you add a letter, it shifts everything over by one, and it makes everything after that mutation completely wrong. That happens sometimes in people. Sometimes it gives like super abilities. Like there's this one weird mutation that a lot of long distance runners have where they alter a protein by altering the reading frame, and as a result they make a dramatically different protein, and so they're more likely to be able to run long distances and not feel tired as they run them. But more often than not, if this happens, it ends up not even being a child who can survive, so it's a miscarriage because there's a gene that was really important that doesn't work anymore. Okay. Mutagenesis is the process that creates mutations in DNA, and a mutagen is anything that increases the risk of mutagenesis. Fairly recently, Johnson's baby powder lost a lawsuit because there was asbestos in it, and asbestos is a known mutagen. It causes mutations to happen in the DNA, and so anytime you expose yourself to things like that, it increases the risk that you get cancer, yes, but also it increases the risk that you can just mutate any gene in your body and end up with problems as a result of that mutation. Okay, so here's what I'm going to do next. I'm going to back out of this, and because we didn't get to do the hands-on things that I usually like to do with this, I have posted a couple of things that should be able to help you out with your DNA stuff, and I'll post a quiz too so that I know you're studying your DNA stuff. So Brain Pop DNA, it takes you to a page where you can watch a movie that helps explain DNA again. You can take some practice quizzes. You can do challenges. You can do like a cognitive map so you understand what connects to what. You can do any of this stuff. Any of this stuff is fair game for you guys, but it can help you learn this, especially if you're struggling with it after the lecture. Also posted in Helpful Quests, I've posted this Quizlet down here. I'll say the same thing for this one as I said for chemistry way back when. If you can beat my time, I'll give you extra credit on the next test, and so play this for a little bit and see how you do. From here, just make sure you take the quiz that I'm going to post. It'll populate off to the side once I get that posted. Good luck.